Hello, everyone. Let's get started. We are going to be doing lecture 3 this class, and your quiz results has been posted. Um, Hopefully, the quiz was what you expected, and there were no surprises. And if you have any questions regarding the quiz, please feel free to send me an email, and I'll be happy to address your concerns. But for now, we are going to move on to lecture 3, which talks about cells. So the first question here asks for the 4 principles of the cell theory. By the way, for any of you who needs to turn on caption, there is an option on Zoom that does that for you. It translates what I'm saying directly into captions. Um, and sometimes I do speak a little bit fast, perhaps, and having those captions might help you. All right, so state the 4 principles of the cell theory. And the first part of the cell theory states that every living thing, every living organism is composed of one or more cells. Okay, so if you're a living thing, then you are going to be made up of one or more cells. Now, some organisms have only a single cell, and we had some of those examples in the lecture notes. So, example, the amoeba, right, is a single cell organism. Um, and for humans, we are multicellular organisms. So humans have trillions of cells. Trillions of cells. So it doesn't matter that you're made up of one cell or many, many cells. You would exhibit the same characteristics of life. So that's the first part of the cell theory. And the second part is a cell. Sorry, I'm just gonna choose the right thickness here. A cell is the smallest unit of life. Okay, individually alive as part of a multicellular organism. Okay, so of course, unless you're a single cell organism, then that's fine. But if you're a multicellular organism, individually, those cells that makes up the whole organism, they are alive on their own. In fact, you can take a cell from your body and you can grow it in a petri dish if you give it the right nutrients, if you give it the right conditions, is able to survive for quite some time outside of your body. And so individually is alive as part of the whole organism. Does anyone want to give me another part of the cell theory? That's okay. I will write the next part then. All cells came from the division of pre-existing cells. Now, no one really know where the first cell came from, but we all know if there is one cell, it's able to divide to make more cells. Okay, so this process is called mitosis, which we'll learn later on. Okay, so this is cell division. You start with one parent cell, and then after mitosis, you're going to get what we call daughter cells. Okay, so all cells came from the division of pre-existing cells. And then finally, last but not least, cells contain genetic information that will be passed down, will be passed on to new cells during division. Okay, so I know these are not exactly word for word what's written in the PowerPoints. I'm paraphrasing and expanding a little bit, but the core ideas are the same. Okay, so there are genetic information in the form of DNA, and these are going to be passed on when the cell divides. You want to make a faithful copy of all the genetic materials. Okay, you don't want any differences, any surprises. All the cells should be identical to the parent cell when they divide. Okay, so there you have it. Those are the 4 principles associated with the cell theory. Okay, next over here, we have fill in the blanks that explain the hierarchy of organization. Okay, you start with the smallest unit, which we learned in lecture 2, the atoms. And we know that the atoms can come together to form molecules. The molecules can be small or it could be big. Right? It's going to become macromolecules. And that's the focus of the second lecture, right? The carbohydrates, the proteins, nucleic acids, and lipids. And after the macromolecules, the next level is going to be the organelles. Okay, just give me a second, guys. I just got an email. I think the student is asking for the link for the class. Let me just reply her. 1 second, please. Okay, and the organelles are basically like tiny structures that we have inside a cell, and they perform different functions. And that would be the focus of lecture 3, of course. And then the organelles, multiple of them anyways, it's going to form the cell. And there are different types of cells in the body that performs different function. And when you have a group of cells that performs various functions that comes together, then we will have tissue. Okay, so let me give you some example of tissues. So we have like connective tissue. Connective tissue helps you glue things together, right? Literally connect different parts of the body together. Does anybody know any other tissue that exists in the body that you might have heard of before? Anything at all?
Soft tissue?
Yeah, there are soft tissues. And then there are like, you know, adipose tissue or fat tissue, right? All these are specialized groups of cells that are designed to perform a particular function. And multiple tissues can come together to form an organ. So we can have like the heart, the liver, the stomach, all these organs are part of a bigger organ system, such as the cardiovascular system, digestive system, and all these systems are functioning together to create the organism. Okay, so in this course, we've done the molecules and the macromolecules already. Organelles are basically today's focus. And the rest of the course, we will be focusing on the cells. There's so much to learn about the cell. How do they replicate? How do they make proteins? And then at the very end of the course, we're going to transition into tissue. And the next semester, when you take the anatomy and the physiology, it's all about learning the organs and the organ system. Okay. Now, there are different types of cells, and they all look different. They might be found in different environment. They might function differently, but there are three common features that are found in all cells. Okay, does anybody remember what are the common features found in a cell? Please do share them if you know what they are.
Cells and membranes.
That's right. So all cells have to have a cell membrane. The cell membrane separates the living environment inside a cell from the surrounding area, which is non-living in most cases. There's another name for the cell membrane. I'm just going to put it here. It's also called a plasma membrane. Another thing, please, other than the plasma membrane.
The nucleus, right? I think that's how I said.
The nucleus, yeah, okay. Good try. But nucleus is not a common feature in all cells. As we will see, nucleus is only found in eukaryotic cells.
Okay.
Yeah, but good try. Anyone else?
Okay, cytoplasma.
Genetic materials.
Yep, so I hear two answers here. There's cytoplasm,
Plasma.
and then there is genetic material. That's correct.
Which is like DNA and RNA.
Exactly, exactly. Okay, so the cytoplasm, it's like the liquid, the fluid that is found inside the cell membrane. Okay, if you think about a Ziploc bag and you fill it up with water, close Ziploc bag, right? And the liquid inside would be analogous to the cytoplasm, and the actual Ziploc bag would be like the cell membrane. Okay, and then we have genetic material. All right, so here is a typical cell. Right, we have the cell membrane on the outside, the cytoplasm, and then the genetic material is going to be like on the inside. Okay, so that's number 3. So these are common features of all kinds of cells. It doesn't matter if you're talking about animal cells, plant cells, bacteria, they all have these features. Now we can classify the cell based on the kind of features it has, and generally we can divide them into eukaryotic cells or prokaryotic cells. Okay, sometimes if you are doing some readings, you might notice eukaryotic cells are also called eukaryotes, and prokaryotic cells are prokaryotes. It's just like shortened form for these names. Okay. So generally speaking, the prokaryotes, they are typically smaller. Okay, smaller in size. They are more primitive. And compared to the eukaryotic cells, they are larger. In fact, some prokaryotes are so small that they could enter into the cytoplasm of eukaryotic cells. You might have heard of listeria. Sometimes you hear listeriosis, contamination in like coquettes products, right? Listeria is caused by a bacteria, and that's a prokaryotic cell, and it's small enough to be able to go into your eukaryotic cells. In terms of the structure, the prokaryotes have simple structure, and the eukaryotes is much more complex. Location of genetic material for prokaryotes, it is just going to be in a central area of the cell. It is not compartmentalized. It's not like sealed off in a separate structure. It's just kind of hanging around in the middle. Okay, so if you think about a bacterium, here's your bacterium. Its genetic material would just be like in the center like that. Okay, so that's the genetic material. Now, what's interesting about the bacterium is that in addition to the central core DNA, it also has some circular DNA. Okay, and these circular DNA, they are called plasmid. Plasmid. Okay, they're circular DNA, and depending on the kind of plasmid it has, it gives them special properties, special abilities. And these abilities often will help them survive better in harsh environments. So the blue plasmid might give it the ability to break down penicillin, right? And so if a person gets infected by this type of bacteria, the antibiotic penicillin will not work for clearing the infection. Okay, and what's scary is that these bacteria can actually communicate with each other and exchange these plasmid. So let's say we have two bacterium, right? You don't have to write this down. You can just listen to me explain it. Let's say there's one bacterium, and then this is second bacterium, and this one has a purple plasmid that allows it to be resistant to the purple antibiotics. And here we have another bacterium that has the blue plasmid that makes it resistant to the blue antibiotics. Okay, and if these two bacteria ever come close to each other, like for example, let's say I am infected with the purple one, and then another person is infected with the blue one, and then we both cough on the same surface, okay? And then these bacteria would now be able to be in close range of each other, and they actually can connect with each other with a structure called a pilus, and they could exchange their plasmid. So after the exchange, then this one would have the purple, and then this one would have the blue as well. Okay, they make a copy of the plasmid and then they give it to each other. So now we got a problem, right? This one, which was originally just resistant to the purple one, is now resistant to the blue one as well. So if I get infected by this, taking the purple antibiotic and the blue antibiotic will not help me, right? So if this process keeps on going and going, right? Maybe this one meets another one, and this one is having like a green plasmid that makes it resistance to the green antibiotics, and then they exchange their plasmid, and now we have, you know, a third antibiotics that's not going to work. And this actually happens in real life. Bacteria keeps on exchanging these material, and eventually we create what's called a superbug. And the superbug is multi-drug resistance. There's no antibiotics that would work to destroy them. And if you get infected by one of these superbugs, your chance of, you know, having a serious infection and dying from it substantially increase. Vicky, you have a question, please.
Yes, sorry. So the location of the genetic material, because I was going through the slides and I didn't see that in the slides. So, and this question came out right now. Are we to expect questions that are not in the slides or in the
So in terms of studying for the test and quiz, it's everything in the lecture slide and everything that I explain during the tutorial as well. So like sometimes in a tutorial, I add in some extra information that, you know, it's related to the slide, but not necessarily on the slide. You are also responsible for them.
Okay, because I didn't see this genetic material location in the slides.
Right.
Okay.
But do you have any question about what I'm explaining? You get it though.
No, I got it.
Okay,
yeah.
Perfect.
Yeah, thank you.
All right, so everybody okay? Like you guys know what superbug is? You probably have heard of it from the news, right? Okay, and these are bacteria that are not going to be able to be killed by a bunch of antibiotics, and as a result, they are super dangerous. You don't want to be infected by one of those. Okay, as for the eukaryotic cells, the location of genetic material is inside the nucleus. Okay, inside the nucleus. Okay, so it's not like loosely hanging around in the center of the cell, but rather here is your cell, and there is a specialized structure on the inside that is specifically designed to hold the genetic material, right? So it's found inside there, and this structure is called the nucleus. Okay, so eukaryotic cells like our cells, they have the nucleus. Okay, there are some exceptions in the body, like red cells, they don't have nucleus, but for the most part, all the body cells that we have contains a nucleus. Okay, number of organelles, few. Right? Bacteria, prokaryotes, they have very few organelles. But for eukaryotic cells, there are many. Examples of prokaryotic cells, I already gave you one, right? Bacteria. Okay, so anybody know any bacteria that you've heard of from the news, from reading the newspaper, internet? Usually people know a few names of bacteria. Usually it's like a letter and then with another name. If I say it, you will know it. Like I guarantee like you have heard of it before. No, nobody knows any bacteria. E something, anyone?
E. coli?
Yes,
E.
that's right.
coli?
See, you guys have heard of E. coli, right? You must have water contamination, right? That's a common bacteria. There are a bunch of other ones, right? Like Staphylococcus, right? That's the one that causes strep throat. If you have kids, you would know about strep throat. Right? Do you have another one? Sorry?
Spirogyra monk.
Yeah. That's another common bacteria. So a bunch of them, okay? But I'm just gonna put E. coli here. So for eukaryotic cells, we have animal cells, okay? Animal cells, plant cells. These are all eukaryotic cells, okay? So they're all bigger, very complex. They have genetic material inside a nucleus. There are many organelles. Okay, so that is that. Okay, let's do a quick poll just to see if you have been paying attention. Go ahead. Okay, I'm just gonna wait a couple more seconds before I close the poll. Please choose something. Yeah, and most
Can
of
you
you.
share the poll, please?
Can I share? I did. Were you not able to see it, Shamila?
No.
Oh, okay. Depending on like what device you're on, sometimes the poll doesn't show up. But that's okay. If you don't see the poll, just make a note of what the answers are, and you can compare it. Okay. Thank you for pointing it out. So the answer is D. Right. All cells is the basic structural and functional unit of living organism. Right? Everything else is not correct. Okay. How about this one? Okay, the correct answer is nucleus. Nucleus is only found in eukaryotic cells. It's not found in prokaryotic cells. Okay, let's see if you know this one. You probably would know it if you've read the recordings. If not, well, actually, we just talked about it too, right? Like in the recordings. I mean, in the tutorial. Yep, the prokaryotes, that's the E. coli, that's the bacteria, right? Amoeba is not a prokaryote, it lives in pond water. That was explained in the lecture recordings. Liver cells, that's part of us, so that's eukaryotic cells. HeLa cells are these cancer cell line that was extracted from a person with ovarian cancer, and the story about that was is explained in the lecture recording as well. Let's see. Yeah, that's it for now. Let's go back to our slide here. We have a picture here to label. This is a mitochondria, and let's just go through some of the parts of the mitochondria. It's a good idea to know the different parts of the mitochondria because in lecture number 5, we will be talking about this extensively. So here, the mitochondria has an outer membrane. This membrane is the same type of composition as a cell membrane. We call this the outer membrane. It's made up of phospholipid, just like the plasma membrane. And on the inside, there is another membrane that's kind of folded up like this. Right? All these foldings actually will help increase the surface area that's available for reaction to occur. When it's all folded up, right, it actually covers more area, and that is called the inner membrane. Okay, I know it's a little bit hard to see, but that's what this line is pointing at. Inner membrane. Okay. So there is some space between the green line and the purple line that I've drawn here, and that's what this is pointing at. Okay, this one is pointing at the space between the outer and the inner membrane, and that is called the intermembrane. Intermembrane space. Okay, so there are reactions that are going on in the intermembrane space, and we'll learn about that later on. And then there is a final line here that's pointing at the center right here, the interior, the inside of the inner membrane, and that is where the matrix is located. And again, there are reactions that are going to be going on in there. Okay. Now, another thing you should know is that the inner membrane folds into what is usually described as finger-like projections called cristae. Called cristae, C-R-I-S-T-A-E. Right? And like I said, the function of these foldings is to increase surface area so that you can have more reactions going on. All right, so there is your mitochondria. All right, let's have a did you know question. Okay. Mitochondria actually has its own DNA. Mitochondria has its own DNA. And these DNA codes for the mitochondria protein. Okay, so I have a question for you. Do you think the mitochondrial DNA, does it come from dad? Does it come from mom? Or does it come from both parents? Let's see what you guys think. The mitochondrial DNA. Which parent does it come from? Or is it both? It's just like, you know, gut feeling. You can tell me, what does it feel like? Okay, that's pretty cool. Most people think it's from both parents, but actually, the mitochondrial DNA is passed down exclusively from mom. Okay, so the mother's mitochondrial DNA gets passed on to both sons and daughters. Okay, and then everybody's mitochondria has DNA that is the same as their mother, and this is the reason. You know, when you have an egg, right, and then there is the sperm. Okay, the sperm is going to fertilize the egg, and the sperm only injects the DNA and nothing else, right? So those are the DNA that makes up half of your genetic material. But then inside the egg, that's where all the mitochondria from the mothers are located. Okay, none of the mitochondria from the sperm gets injected, and so these are the mitochondrial DNA that gets passed on to the next generation. Okay, so they actually can use mitochondrial DNA to trace lineage in some cases. Anyway, that is that. Any questions so far before we move on to the next section? All right. Next, we have described the functions of each component of the endomembrane system. Okay, so let's quickly define what an endomembrane system is in case you need a refresher. So endomembrane system refers to a series of membranes inside the cell. Okay, and they have interrelated functions. Okay, that's what endomembrane system means. Endo means within, right? So it's a network of membranes that are within the cell membrane, and they have interconnected, interrelated functions. Okay, so I'm just going to explain this in a broad sense, and I'm going to come back to this chart. All right, feel free to draw along if you want. If you have watched the recordings already, maybe you can just sit back and watch me do it. Okay, so here is the center of the cell, and this circle represents the nucleus. Okay, so the nucleus is actually surrounded by an envelope, a membrane, okay, that goes around it. Okay, like that. But this envelope not only surrounds the nucleus, but it actually continue to extend outward into the cytoplasm. It continues to extend out like this. Okay. So this is the cell, right? This is the nucleus. And then I'm telling you that The nucleus is surrounded by the membrane, the nuclear envelope, and then it actually extends out. Okay, I'm just like zooming in here to make a clearer picture, right? Okay, so we'll come back to this thing later, but let's focus on the envelope here. So that is the nuclear envelope. Okay. Now, the envelope has holes on it. Right? So like there are some gaps here and there. And these gaps allows things to go in and out of the nucleus. And some things need to go in, like hormones, something needs to come out, like ribosome, which we'll talk about later. But these gaps, they are called nuclear pores. Okay, so allow things to go in and come out. Right? Various things can enter or leave the nucleus through the pores. Okay. Now, on the inside of the nucleus is another circular structure. Okay. Does anybody remember what that is called? The best way to think about this is like eating a peach. Okay. The thing in the center, that's like the seed. Okay. And then the skin of the peach is like the nuclear envelope. Anyways, what is that thing called? Anyone at all? No, that's fine. That is the nucleolus. Nucleolus. Okay. You guys haven't watched the recordings yet, right? Have you? Is that why a lot of you are very quiet? I guess it's busy, right? With a lot of quiz and tests going on. Yep, I just checked. Only 5 of the 31 students have watched the recordings. That's okay. Make sure you do watch it though, okay, after class, to learn everything else, okay? So that's the nucleolus, and this is where you produce something called ribosome, which we'll get to in a second. Now, in addition to the nucleolus, you will also have some genetic material right here. Okay, so this genetic material is called chromatin. Okay, and that is DNA wrapped around proteins. Okay, the DNA is too long to pack inside the nucleus. Right? It's gonna get tangled up. It's gonna, maybe break if you try to shove it in. And so you have to wrap them around these special proteins, so they're more compact and you can store them easily. After you wrap them around proteins, the final product is called chromatin. Okay. So that's that. Now, what happens next is the nuclear envelope is going to extend into the cytoplasm and form this network of membrane. Okay. And on these membranes are going to be another organelle, right, called the ribosomes. Okay, so I already told you the ribosomes are made from the nucleolus, and once you've created them, they will exit through the pores, and some of them will attach to this network of membrane. So with the dots on them, this stuff has a rough appearance, and that's why they are called the rough endoplasmic reticulum. okay, or RER. And the function of the rough endoplasmic reticulum, and I'll write down in the chart later, is to produce proteins. Okay, when they produce proteins, the proteins will be packaged into a vesicle. Okay, so like imagine proteins are being created here, and then this part of it will break off, and then it will become a vesicle. So there's a vesicle, and inside the vesicle, you will have some proteins in it. Some proteins. Okay. I'm going to use a slightly different color here to represent something else. Some of the network will have the ribosomes on them, and we call them rough endoplasmic reticulum. But the network will continue to extend. And then you will have another network out here. Okay, and these networks do not have the ribosomes on them, and they have a smooth appearance. And so instead of calling them rough endoplasmic reticulum, these are called smooth endoplasmic reticulum. Okay, which we'll come back to later on. So there's one final thing here that's part of our endomembrane system, and it's something called the Golgi body, Golgi apparatus. Okay, and it's a bunch of membranes that are stacked together, kind of like pancakes. Okay, so this stuff is called the Golgi body. And the purpose of the Golgi body is actually to receive the vesicles that was coming out from the rough ER, and it will sort the contents of the protein. It will modify it if necessary, and then it's going to repackage it into a new vesicle. Okay, let's use a different color to represent a new vesicle. New vesicle, and then, you know, the protein like that. So this is a new vesicle. The contents might have been modified a little bit. Okay, and that's essentially the endomembrane system. Okay, it's a network of membranes inside the cell membrane, and their functions are all interconnected. They're related to each other. Does anyone have any questions before I go back to filling in the chart for you? Okay. So here we go. Let's fill in the description and the function. I will use maybe pink for let's just use red, I guess. Red for description, and then function I'll use blue. Okay. So on the next quiz, which is quiz number 2, and it covers lecture 3 and 4, you will have some of these diagrams that you need to label. Okay, so I will show you this thing, and you have to tell me what it is. Okay. So this is pointing at this structure right here. This structure, as opposed to out here, and you can see all these black dots on them, right? Okay, so that tells you this is the rough endoplasmic reticulum. Okay, RER. And so let's have a bit of a description to it. It is, as I told you, continuous. Continuous with the nuclear envelope. Okay. And it's called rough ER because it is covered covered with ribosome. And the ribosomes are made in the nucleolus, right? So what does it do? The rough ER and ribosomes work together to produce proteins. Okay. And like I showed you earlier, proteins are packaged into vesicles that buds off from the rough ER. It's kind of like a bubble that pinches off from the rough ER, and then that vesicle is gonna go somewhere else, right? Does anyone remember what that something somewhere else is? I literally just told you in the drawing. I just want to see if you remember. Where does the vesicle go after it comes off from the rough ER? No? That's okay. It will go to the Golgi body, right? So it's going to butt off from the rough ER into a vesicle, and the vesicle will go to the Golgi body. Maybe we'll write something about that in a bit. Any question about the rough ER before I move on to the next thing on the list? Alrighty. Next part is the over here. Right? That's the smooth ER. Smooth endoplasmic reticulum. Okay, so let's do the description first. They're continuous with rough ER, just further out from the nucleus, and they are not covered by ribosome. That's why they have a smooth appearance. And the function of the smooth ER is the synthesis. Synthesis means to produce, to create, right? Synthesis of lipids, such as steroid hormones. You might remember studying for those, right? The testosterone, the progesterone, those are lipid steroid hormones. And so in the ovaries and in the testicles, the cells that makes those hormones, they would have a lot of smooth endoplasmic reticulum. Okay. So expect to have a lot of smooth ER in ovarian cells, right? Because that makes the sex hormones. Right? They also play an important role in detoxification. Detoxification of lipids and drugs, like medications that we take needs to be metabolized, right? And the smooth ER is going to do that. So what organ plays a role in detoxifying drugs? Do you know?
The liver?
The liver, that's right, Kamon. And you would therefore expect to have a lot of smooth ER in the liver cells as well. Okay. So that is that. Moving on, we have a stack of these membranes that are formed in close proximity. We call these the Golgi body. Golgi body, or in some older textbooks, they are called Golgi apparatus. I'm just telling you in case you run into it and you don't know what that means. Okay, it's the same thing. Okay, so it consists of layers of membrane that are located very close to each other. They're not necessarily touching, but they're really close to each other. And that vesicle that came off, right, from the rough ER, that's gonna go to the Golgi body. Like this guy that buds off from the rough ER, it's gonna travel to the Golgi body, and it's gonna enter the first stack, and then it will get sent to the next stack and the next stack, and sometimes they go backwards, back and forth a couple times before finally coming out as a new vesicle. Okay, so the function of the Golgi body is to sort, modify, and repackage proteins that are in vesicles. Okay, and just want to reiterate that these vesicles came from the rough ER. Okay. Are we okay? I know it's a lot of information.
Yeah. Yeah.
Okay, thank you for confirming. I hope everyone else is okay. Just gonna go back to this drawing here, okay? Now this new vesicle has a couple of possible destination. Okay, some of it is actually going to become like a specialized type of organelle called the lysosome. Okay, so it could become lysosome, which I'll explain in a bit. It could become just a vesicle that is being stored in the cytoplasm until the contents are needed. Okay? Not every protein that you make is needed immediately. Okay, sometimes if you have extra resources, you would like to make some of them and just store them there, and you can release them on demand when you need them. Okay, so some of them is just going to hang around in the cytoplasm, but sometimes you need to make them and need to send it somewhere else. So the vesicles will release the contents, the proteins, out of the cell. Okay, and sometimes the proteins will become part of the membrane. Okay? The protein that you make becomes part of the membrane, and that's something that we will explore more next time. But my point is, there are various destination for the vesicle. Right? It could become one of any of these scenarios. As far as the table goes, we're gonna focus on this thing called a lysosome. Okay, lysosome. So that's what the lysosome looks like. Lysosome. So it's a small sphere that contains digestive enzymes. Okay, you know what enzymes are now, right? There are things that speed up chemical reactions, and in this case, these digestive enzymes will help you break things down. So what kind of things are we breaking down? The lysosome is responsible for breaks down macromolecules and things the cell engulf. Okay. Not all cells are going to be like eating things, but some cells, like our white cells, regularly eat up things, eat up bacteria that's not supposed to be there, and then you need to break them down, right? That would be the function of lysosome. In some cases, they are also important. The lysosomes are also important because they triggers programmed cell death. Okay, this has a name. It is called apoptosis. Okay, so when do you want a cell to die? More often than you would expect. A cell, if it's too old, if it's worn out, if it's infected, right, then you want to get rid of it. And so the lysosome is going to cause the cell to basically destroy itself. We call that apoptosis, or programmed cell death. During embryonic developments, right, like our fingers are not actually separated. There are webs between them, and those cells need to die. And so to kill off those cells, trigger apoptosis. And that would be the job of lysosome. Okay, so everything that we described so far is part of the endomembrane system. Okay, these four things: the rough ER, smooth ER, Golgi body, as well as the lysosome. Yeah, any question? Now, in the lecture notes, I also talk about how depending on which enzyme is missing, sometimes in certain genetic condition, the enzymes that are in the lysosome might be missing. And depending on which one is missing, it could lead to a bunch of conditions. And we talk about two examples in class. I'm just gonna put them here, and you can watch the recording to get more information on them. One of them is called Pompe disease. Okay, and in this case, the enzyme that's required to break down glycogen is missing, and as a result, glycogen, which is something that we learn, right? It's a storage form of energy, but if you have too much of it, the glycogen will accumulate, builds up, right? Accumulation in heart muscle. And all the muscles in the body as well. But particularly if it accumulates in the heart, then it's going to cause cardiovascular problem. Okay. There is another one that you might have heard of, is called the Tay-Sachs disease. Tay-Sachs disease. And this time it is an accumulation of lipids, because you don't have the enzyme to break it down. So the accumulation of the lipids in brain is going to lead to developmental problems, right? And it could potentially be lethal as well. So depending on which enzyme is missing, there's a whole list of conditions that are associated with lysosomal enzyme, right? We collectively call these lysosomal diseases. Okay. All right, let's do some more polls here. Go ahead, please. Okay, anyone else? The nucleolus produces ribosome actually. Okay? The control of the movement in and out of the nucleus, that's more like the nucleopores, right? If you look at the picture that we've drawn here, right? The nucleolus is the inside the nucleus, and that produces the ribosome. The nucleopores, that's where things goes in and out of the nucleus. Okay, why don't we take a quick break, 5 minutes, and then we will fill in the rest afterwards. We will resume at 1:38. See you in a bit.
Okay, thanks.
Thank you. All right, let's keep going. So moving on, the next part here, we are asked to list 3 organelles or structure that are unique in plant cells and briefly describe their function. I do want to change this a little bit because I realize it's a little bit misleading here. Instead of saying unique to plant cells, we would like to look for 3 organelles or structure that are in plant cells and not animal cells. Okay, because some things are found in plants and other stuff, but not animals. So since most of you haven't seen the lectures yet, I'm just gonna tell you what the answers are. Okay, so there is something called chloroplast. Okay, so the chloroplasts are found in plant cells but not animal cells, and the function of the chloroplast is that it contains chlorophyll that allows the plant to perform photosynthesis. Okay, and that's of course taking the energy from the sun and then using it to create food, right, in the form of starch. That is the function of chloroplast. Another feature that is found in plants but not animal cells are cell walls. Okay, cell walls. So this is made up of cellulose, which we learned in lecture 2, that provides structural support. And prevents the cell from bursting. It's like having an extra layer of protection. So like, you would have your plant cell here. That's the nucleus, and outside of the cell membrane, you would have the cell wall. Okay, it's an extra layer of protection. That's a cell. So that's a nucleus. And in addition to the nucleus, much of the cell, plant cell, is actually taken up by this huge structure called a vacuole. And that's our last item here, a central vacuole. Okay. And the purpose of the central vacuole, for the most part, is responsible for storing water. Stores water and nutrient. But in some cases, depending on the species, the type of the plant, they can also store poison. and pigments. Pigments are like colors, right? So these are the three things that are kind of unique, right, to the plants in the sense that they're found in plants, but not in animal cells. Okay, but it's possible to find cell wall in like fungus, for example. It's possible to find chloroplast in some pond living organism. Okay, it's just that they are not in humans, basically. Okay. Moving on, we have a bunch of other organelles here, starting with this thing, and we know it is the nucleus. Okay. So in the center of the nucleus, that thing, that is the nucleolus. Nucleolus. Now, I drew this out earlier before the break. There is some genetic material here, and that is in the form of chromatin. Okay, so just to recap, you have your DNA, your double helix, right? DNA, which is really long, and you can never fit it into the nucleus. So you're gonna add some packaging proteins. Okay, they're like little circular proteins that you can wrap the DNA around. Think of like a yo-yo, right? Yo-yo has the long string and then the ball at the end, and you can coil them up. Okay, so imagine like you have not just one yo-yo, but like a bunch of yo-yos. And then, you take your DNA and then you wrap it around them, and then you wrap it around the next one, and wrap it around the next one, and the next one, and so on and so forth. So you can compact them nicely, right? And that will form your chromatin. Okay, so they're packaged nicely, right? So they don't really break. Okay, and then you have these proteins that are around them, right? Just like the yo-yo analogy I told you about. But if you have seen in like sci-fi movies or like even any kind of TV stuff now, right? If they want to show you a DNA, you probably see DNA like in this particular form, right? Okay, so this people say chromosome. Right? So the chromosome is essentially taking the chromatin and then condensing it even further. Right? So you take this thing and then you coil it up even further. Coil. And then in the end, you have your chromosome. So essentially the DNA and the chromosome, they are the same thing. It's just like how compact is it? Like how much did you coil it up? And the most condensed form is called a chromosome. Okay, and that only happens when you are trying to, divide your cells. For the most part, in the nucleus, it exists as chromatin. Okay, so description, right? The nucleus is found in eukaryotic cells, right? Not in prokaryotes. It is surrounded by nuclear envelope contains nucleolus. In terms of the function, it protects chromatin. Okay, it seals it off nicely. And the nucleolus produces ribosome. Okay. So next up, we have some ribosomes here. And some ribosomes are bound to endoplasmic reticulum. Okay, some are bound to rough ER. Others are freely floating in cytoplasm. Not all of them needs to be stuck to the rough ER. Some of them are just like floating around on their own in the cytoplasm. Okay. If you take a closer look at the ribosome, right, it's actually made up of two parts. It's like a burger. Okay, it has a top part and then it has a bottom part. Okay, so this is called a large. This is called a small subunit. Okay, so it's made up of large and small subunit. That contains proteins and RNA. Right? It turns out the ribosome themselves actually is made up of protein and RNA. And the function of the ribosome is to produce protein. Right? Can either produce protein along with the rough ER, or it's able to produce it on its own in the cytoplasm. Okay, so that's the ribosome. Now, in the lecture, we talked about something called the central dogma of biology. Dogma means like the central theme, right? Central idea of biology, which is the flow of information. We start with DNA, okay? And of course, we found the DNA in nucleus. The DNA is too precious to be taken out of the nucleus. And so if you want to get some information from the DNA, we're going to have to make a copy of it in the form of RNA. So this process of making a copy of the DNA in the form of RNA, this is called transcription. Transcription. And once you make the RNA will exit the nucleus from the pores, right? And exits the nucleus and goes into the cytoplasm. Okay, goes into cytoplasm. And in the cytoplasm, you are going to have the ribosome, which will decode the RNA and it will make it into protein. So this process of taking the instruction in RNA to make protein, that is called translation. Okay, and it happens in the cytoplasm. In cytoplasm, either you can do it with the rough ER or just the freely floating ribosomes can do it. The transcription happens in the nucleus, right? Nucleus. Transcription in nucleus. Right, so this central theme represents how information is encoded in the DNA, and then in order to extract that information, to use that information to make protein, we have to go through two steps, transcription and translation. So here is our ribosome. And you're gonna have a piece of RNA that you've copied from your DNA. This is the RNA. That purple thing here is the ribosome. And as the ribosome reads the information of the RNA, it will know what amino acids to link together. right? And so here you're gonna build your amino acids, connect them together, and that's how you make the protein. Okay, so from DNA to RNA and then to protein. Central dogma, central theme of biology. Okay. All right, moving on. Next, we have this thing, which you should recognize now. We labeled it earlier today. This is the mitochondria. Mitochondria. I'm not going to go through all the details again, right? See question. What was the question? Question 6, question 5. See question 5 for structures. Okay, the outer membrane, the inner membrane, and all that stuff. And in terms of the function, right, it plays a role in cellular respiration. Okay, and we learned about cellular respiration in lecture 2, right? It's the key chemical process by which we make energy. So it involves taking glucose, C6H12O6, reacting it with oxygen, and then producing carbon dioxide, water, and energy in the form of ATP. Right? So one glucose will react with 6 oxygen, producing 6 carbon dioxide, 6 water, and up to 36 ATP. That is the main function of the mitochondria, to allow this process to occur so we can form energy. Okay, usable energy for cell. Okay. Next here, we have a pair of these kind of pasta looking thing. They are called centriole. Centrioles. Okay, usually they exist as a pair. Exist as a pair. And usually arranged perpendicularly. To each other. Okay. And the function is to produce something called spindle fibers. That help guide chromosomes during cell division. Okay, the chromosome needs to know where to go, right? What moves this way, what moves that way, how many should go to one side, and then how many should go to the other side. All these movements are not randomized. All these movements has to have something to guide them. And that's the function of the centrioles. Okay, they send out these guiding lines called the spindle fibers, and it will move the chromosomes for them. Okay, so I should put a star here. Only in animal cell and not plant. Okay. Next, we have these little hair stuff. They are called cilia. Cilia. Okay. So cilia, they are like hair-like structure that allows, that usually covers the entire cell. Okay, so it could be used for movement, or in some cases, or moves things around. Okay, so what do I mean by that? In the lecture note, I show you this single cell organism. That looks like this. And this is called a paramecium. Okay, and it has cilia all around it. Okay, so when these cilia moves, it helps the paramecium to swim around in the pond water. So that's used for movement. In the human bodies, like the fallopian tube. That's ovary, and then there are these tubes that connects to the uterus, right? Uterus. That's where the baby grows. So this tube here, the fallopian tube, the inside is covered with these cilia. So when the egg comes out of the ovary, the cilia will help guide the egg towards the uterus. Otherwise, the egg doesn't really know where to go. So the cilia kind of swoop them along, and that's what I mean by move things around. Okay? It doesn't have to be the cell that's actually moving. It could be it's helping to move something else. In this case, helps to move the eggs. Okay. And this last one here, it's talking about the tail, and that's the flagellum. Flagellum. Okay, and the purpose of the flagellum, well, first of all, the structure is usually just one or two at most per cell. And it's for movement. So in humans, there's only one example of where there is a flagellum, which is the sperm, helps them swim around. But some organisms, they have two. Okay, so like some cells, they have two flagella, and that helps them swim around as well. Okay, but in terms of human, just one, and it's only found in male, obviously. Okay, so that is that. See if there's any more poll questions that we can do. All right, let's see if you were paying attention. Go ahead. Okay, well, thank you for trying. The answer is protein and RNA, right? We wrote this down in the chart. Those are the key components of the RNA, the ribosome. Okay, how about this? Okay, I only have 7 responses, 8 now. Again, I'm just curious, is it because you guys don't see the polls, or is there other reason that you guys are not participating? I'm actually gonna have a little survey that I'll send out to you after class, just to, now that we have done 3 weeks of this, I want you to give me some feedbacks as to, what kind of changes, if any, you would like to see in the
Please,
course.
I don't see the poll.
Oh, you don't see the poll. Oh, okay. And
Yeah.
are you using the phone or are you using the computer?
I'm using a laptop.
Oh, that's interesting. Okay. Maybe other people are in the same situation as you. Well, thank you for letting me know. I'm not sure how to fix that though. But in any case, the correct answer for this would be breaking down macromolecules, toxin, and waste. So lysosome is part of that, right? It has different enzymes to break down macromolecules. But smooth ER also plays a role, right? Smooth ER, we wrote it down over here. The smooth endoplasmic reticulum, detoxification of lipids and drugs, right? So those are the answers. Everything else is not involved in any of these processes. Okay. This one you probably won't be able to do right now because you need to watch the lecture recordings to find out. But I sort of wrote it down for you, right? Like the destination of the vesicle, right? Some of it is going to go to the membrane. Some of it will be in the lysosome. Some of them will be freely floating in the cytoplasm. And this is the exception. Okay, peroxisome is it's something else that also involves in breaking things down, but it uses oxygen radical to do it, rather than using protein. So that would be the answer. When you revisit this question after you've watched the lecture recording, it will make more sense. Okay. All right, let's do another one here. I actually reviewed a lot of your quizzes, and just to see where people are getting their answers wrong. And it turns out a lot of you are struggling with the multi-select questions where you have to choose multiple things. Remember, you must choose all the correct answer in order to get full marks, right? If you choose extra answers, or if you're missing answers, then you will lose some marks for that as well. And when you type your answers in those written response questions, like the question I asked you to write the hypothesis and stuff like that, a lot of you just type me single word answers, and that's probably why you didn't get a full marks. Okay, so make sure you be a little bit more specific in answering those questions. In any case, this is an example of a multi-select question, right? False statement. Smooth endoplasmic reticulum is part of the endomembrane system. That is true. That's correct. We're not gonna choose that one. Oops. What happened there? We're not gonna choose that one. There is an abundance of mitochondria in muscle cells. That makes sense because the muscle requires a lot of movement, a lot of energy, and mitochondria will supply that. So that's correct. We're not gonna choose it. All cells have cell walls to protect it from the environment. That's not correct. Only plant cells have cell wall. And so this is false. Hydrogen peroxide is found in lysosome. That is also false. It is found in peroxisome. Okay. Chlorophylls are found in chloroplasts. That is true. So the incorrect statements are C and D. All right, let's finish this up. Did that already. Now we're just gonna fill in this chart over here. Directly involved in protein synthesis. That would be ribosome. The region inside the cell, except for the nucleus, would be the cytoplasm. One of the common features that you will find in all cells. Organelles that manages or control all the cell functions in the eukaryotic cell, that would be the nucleus. Digest excess or worn out cell parts, food particles, and invading viruses or bacteria. That would be the function of lysosome. Small bumps located on portions of the endoplasmic reticulum. That would be the ribosome. If you have ribosomes on the ER, we call them rough ER. Involved in detoxifying harmful substances and digestion of fat, produces hydrogen peroxide as a byproduct. Okay, the hydrogen peroxide is what gives it away. This is talking about peroxisome. Produces a usable form of energy for cell. That would be the mitochondria. Sorts and modifies contents of vesicles. That is the Golgi body. Right, the stacks of membranes, right? You might remember. Site where ribosomes are produced. That would be the nucleolus. Provides support for the cell. That would be a group of fibers, which we call cytoskeleton. I did not go through it in this tutorial, but it's in the lecture notes. Small hair-like structure used for movement, cilia. Forms a selectively permeable barrier around the cell, plasma membrane. Long whip-like structures used for movement. That would be the flagellum. Contains chlorophyll needed for photosynthesis. That would be the chloroplast. Large sac filled with water in plant cells. That would be the central vacuole. Produces microtubules fibers needed for guiding chromosome during cell division. That would be the centriole. Those yellow pasta looking things. Okay. So you should create like cue cards probably, right, to help you remember what each one of these things do. Describe the key events involved in production and trafficking of substance in the cell. I already drew a diagram that explained this, right? It's like this one that we drew earlier, so I'm not gonna redo that. These two are very self-explanatory. You will find the answers and in the lecture notes, and I'll post the answer as well, right, to the study guide. Okay, let's go through these application questions. Speculate what will happen to an animal cell if it is placed in pure water. Do you expect the outcome to be different for a plant cell? Okay, so let's take a look at number 1 here. if you have a beaker of pure water and you put an animal cell in there, okay? Anybody want to speculate what happens before I tell you the answer? What's going to happen to our animal cells here if we put it in water? Well, the water is going to enter the cell because there is more content in the cells than the surrounding area. So the animal cell will swell and burst, okay? But not for the plant cell, because the plant cell, as you remember, has extra protection. There's a cell wall around plant cell, right? So cell wall protects the cell from bursting. Okay. Next, number 2. Your heart muscle needs to function continuously throughout life. Which organelle would you expect to be the most abundant in the heart cell? So we did a multiple choice question earlier for this, right? You would expect to have a lot of mitochondria, right? For continuous energy supply, right? And we have number 3 here. Endocrine glands produce and secrete hormones. Which organelle would you expect to be most abundant in cells of an endocrine gland? Justify your reason. So what does everybody think? If we need to secrete hormones, what kind of organelles do we need a lot of? Anybody? Okay, well, it depends on what kind of hormones we are making. Right? Remember, hormones can be lipid hormones, or it could be protein hormones. Okay. So if we're making lipid hormones, then you would expect to have a lot of smooth ER. The smooth endoplasmic reticulum is responsible for making lipid, right? Well, and steroids. So if it's steroid hormones that we're making, then you would have a lot of SER. If it's protein hormones that we're making, like insulin, for example, you would expect to have a lot of rough ER and Golgi bodies. Okay, so this is a little bit of a tricky question because you have to think about extra things, right? Like what kind of hormones are you actually making? And depending on the answers, you're gonna have to give 2 different scenarios. Okay, so that is it for this week. Just hang on a second. I'm gonna pull up the class schedule and just see if there's anything critical that's coming up. So let's see. Next week we will do lecture 4. Okay, so nothing special there. And then the week after that, you are going to be doing your lab. Okay, so I might need to spend some time to talk about the lab with you next class as well. Okay, but we'll see how it goes. Okay, but make sure, please watch the recordings before coming to class. It will help you understand the stuff much better. All right then, you guys stay safe out there. It's like a blizzard, and I'll see you next Wednesday afternoon. Have a good week, guys. Bye.
Thank you. Bye.
Thank you.